So carbohydrate digestion happens in the small intestines. We get the process started a little bit in your mouth in the salivary amylase, and we continue the process with pancreatic amylase. It's really the same amylase, but the problem is in your mouth, the salivary amylase only gets activated from your mouth to your stomach and sort of denatures in your stomach acid. Once we're done with that, we're going to go ahead and when the food comes out as chyme, we're going to have to make another amylase, and that's going to be pancreatic amylase. And that's going to be acclimated to the pH of 7, and it's going to do all of its job in the small intestines. We have other things that are going to help us break down these sugars, and go ahead and refer back to your sheet for all of them. But we're going to have disaccharides, which are going to be these large sugars like sucrose, lactose, and maltose, and they're going to need an enzyme to break those down. Sucrase, lactase, maltase to help break those down. Polysaccharides, which are just really long strings of sugar, are going to get broken down into monosaccharides and be absorbed into the body through other enzymes. And then, of course, we have the enzymes that we just talked about. Carbohydrate absorption is basically going to be moving the sugars from the small intestines through facultative diffusion. So diffusion through a special protein that's going to move it from high concentration to low concentration, and it's just going to keep moving it that way to move to the other side. So glucose and galactose sometimes are needed because we're going to move bulk amounts of it through, and we're going to use the secondary active transport in the lumen, and that's going to facilitate diffusion through the epithelial cells and sort of dump them into the bloodstream a little faster. But usually fructose is the only one that we'll do, or one of the few that we're going to do just facilitated diffusion. Everything else is going to use some sort of secondary mechanism. Anywho, we have our sugar, and we're going to move it through the small intestines, and we're going to dump it into the capillaries, the blood system. We've already talked about how proteins move through, so let's talk about how fats move. Lipid digestion is going to be a little harder to do because fat wants to sort of conglomerate with each other and sort of become this large material, and that will become very difficult to move. So we have this sort of kind of complicated mechanism that we use to take that fat and sort of emulsify it to become easier to move around, and to make it so it doesn't go back into this big fat globule and then clog the small and large intestines. That would be terrible. What we're going to do is we're going to produce bile salt in the gallbladder, and it's going to get released every time you eat a little food into the small intestines. That's going to help emulsify, which means break down fat molecules, so it's going to be a little easier to move. And then pancreatic lipase is an enzyme that's going to break those fat molecules in the small intestines and move them to the lacteals. We have to be a little specific with this one because pancreatic lipase is what we have to call it, because kids will make a lipase in their mouth, babies, when they're breastfeeding. And once they stop breastfeeding, they stop making that one, and this is the one that the majority of the people make. Lipid absorption is going to just happen as the fat gets broken down into the small intestines. We add the bile salts. It's going to make those things even smaller. Eventually those villi and the microvilli will move them to the lacteals, and the lacteals is where we're going to dump them either into the bloodstream or store them in those adipose cells. And what's going to end up doing that is these lipoprotein lipases, which are associated with that capillaries, and they're just going to move them to the lacteals to help them diffuse that direction. So how that works is, here's your bile salts added to make them small. They're going to go through using simple diffusion right into the lacteals and sort of get deposited into the lymphatic system. And of course, we have all of these vitamins and nucleic acids, and they're all going to get broken down in the brush border. So we know that nucleic acids are going to get broken down by nuclease, and that's going to break down DNA and RNA to those A's and T's and C's and G's that we can use to build our DNA later. We're also going to be able to move vitamins through that system, and that's going to go ahead and make the vitamins sort of come out and be either stored in the liver until we need it, or cells will just automatically just pick them up and sort of move them in. And then I think that's all we have. So happy studying.